This is nice to follow Dom and Bob and let you know a little bit from the nucleic acid side. The work that IDT has really done, especially from the R&D group, for probably the past 20 years or so. I've been at IDT for 10 years. During that time, worked a lot on siRNA screening, dicer substrates, chemical modification of those siRNAs, as well as antisense oligos. And then in the last 5 years, I think our functional genomics team, as Bob mentioned, has turned almost wholly to CRISPR development. We saw an opportunity as a nucleic acid synthesizing company to make a play in this space, and Ashley will talk more about those specific products a bit later. So I'm going to focus on kind of what is the old days of the work that we had done in our functional genomics group. But as Dom mentioned, this is a little bit resurgent now, or a lot resurgent now that there's an FDA-approved antisense oligo. And there are some other factors, too, with the availability or off patent of some 2' modified RNAs that enable our opportunity to further improve these technologies. So I think this is probably pretty basic, but I want to start here just to let you know that we have solutions for RNAi with siRNAs. We've done, as I mentioned, a lot of work, a lot of screening, a lot of understanding of chemical modifications for knockdown of target mRNAs with siRNAs or our dicer substrates, as we call them, that induce a polarity. I can talk more about those if you are interested in understanding more, but they're more on-target specific than the standard Tuschl siRNAs. We've also done a lot of work with antisense oligos, both targeting mRNA via RNase H1 mediated knockdown, as well as looking at the steric blocking ability of those antisense oligos. As you might know, this is the Onpattro type of mechanism that is being used in that FDA-approved drug, blocking some mis-splicing of that mRNA and then misfolding of the protein. Getting a little bit more into the nitty-gritty of what exactly these IDT tools look like in terms of these applications. Typically, our screening duplexes for siRNA applications, our dicer substrates are fully ribonucleic acid, except for the 3' end of the sense strand that has 2 DNA bases to stabilize, and you'll notice that there is a polarity, a 3' overhang on the antisense strand, rather than, as I mentioned earlier, the 21 mer designs that are the Tuschl patented original designs. We've shown a paper in 2005, in fact, that these are more potent, more specific than the 21 mers. Oftentimes, when it comes to RNase H active antisense oligos, there's a requirement of a DNA stretch, and say often these are thought of as 20 mers, but recently with the development and the availability of locked nucleic acids that can be present in the wings, the gap mer notion is a requirement for RNase H1 activity because RNase H1 cannot cleave the 2' methyls, cannot cleave the LNAs, and so in the center of those specific oligos targeting the mRNA is required that they contain DNA bases. But what I was mentioning is that the Affinity Plus version or the LNA version or a Mo modified version indeed can be shortened to 16 mers and retain the same potency as the 2' methyls. And then the steric blocking oligos, just to shortly mention them, I won't show any functional data when it comes to these, but these can be fully modified and fully phosphorothioated throughout. And then just to get into a little bit of some of these chemical modifications, again, as I mentioned, these are 2' modifications on RNA. The 2' O-methoxyethyl, or the 2' Mo, as we call it, is something that's newly available to IDT. These indeed, and I'll show some data, are stabilized against endo and exonucleases. They are a bit bulkier than the 2' O-methyls and may relate to some of the reasons why they're more effective in making and stabilizing those antisense oligos. And One thing that's maybe key to mention, but maybe not relevant to this discussion, is that these are amenable to gymnotic or naked delivery as well. There are some reasons why that might be an advantage in some cases. But as I mentioned, that these are not a substrate for RNase H1, and so that they need to be present in the 5' and 3' ends and make that middle region available for DNase cleavage or RNase cleavage with the DNA. Additionally, and similarly, the 2' methyl is often used and is a bit of a cheaper option than the 2' Mo, and so for some screening applications, this can be quite useful as well. And then I just want to mention the backbone modifications that we often use, and I'm just kind of highlighting certainly there are more chemical modifications available from IDT, but when it comes to ASOs and siRNAs, these are typically those that we think of. And Ashley will mention some of these as well in the chemical modifications of our CRISPR RNAs. But often the phosphorothioate modification is one that we use to further stabilize the ASOs. And then to actually show some functional data, I mentioned that the 2' methyls may be more of a screening application. We see across this genomic or this mRNA for beta-catenin, I almost started to say genomic locus because I'm so used to speaking about CRISPR applications that I have to reel myself in here a little bit. But the 2' methyls don't quite give the same potency as do the 2' Mos, as do the as do the LNAs. You see that represented in this figure for each of the sites. Oftentimes at both doses, this is a liposuction in tissue culture, that at the 10 and the 3 nanomolar doses, the LNA provides the most knockdown, the greatest knockdown. However, there is an issue with toxicity associated with the LNAs, and then that's why the 2' Mo modification has turned to be one of our favorites when it comes to the modifications of the antisense oligos. So this would be the type of screen that we would go through and look for lead candidates if we were working collaboratively with an investigator to try to come up with the best antisense oligo that could, for this example, target beta-catenin. Similarly, we've done quite a bit of work in the past looking at many different, these individual dots all the way along the x-axis represent different siRNAs or dicer substrate targets across a gene of interest. All I want to do is to show here that we do indeed have smaller scale dicer substrates for screening activity. A lot of the work that we had done in the past to develop lead molecules that would be clinically targeted for anti-cancer or metabolic disorders were done in this way, in which we initially did a large screen, looked for the most potent siRNAs, underwent then further chemical modification studies to make sure that we held potency while further stabilizing the dicer substrates with most commonly 2' methyls. And then an application in the last few years, Kim Lennox led this project really in our R&D team, in which she looked at the localization and the knockdown and the ability to knock down long non-coding RNAs. So this is no longer looking at messenger RNA, but again, single-stranded RNA or long non-coding RNA. She has 2 nuclear targeted or nuclear localized long non-coding RNAs, which are mallet1 and neat1, and then dancer and OIP5 are 2 cytoplasmic targets. And I think you can see from the comparison of modalities here of knockdown that indeed the antisense oligos, or the 2', specifically the 2' methyl gapmers, in this case they're 20mers, are more effective knocking down the nuclear localized mallet1 and neat1, whereas the RNAi or the dicer substrate application is more effective because of the location or the prevalence of the dicer and RISC machinery in the cytoplasm in knocking down those cytoplasmic localized targets, dancer and OIP5. And then nicely, this final figure shows the combinatorial effect that can be achieved combining the antisense oligos and the dicer substrates to give across the board, both for nuclear and cytoplasmic or co-localized long non-coding RNAs, the most potent knockdown mediated by incorporating both of these. One other one other modality that I haven't really touched on recently, and we haven't worked with too much, but there's been a little bit resurgent because of the 2' Mo modification, has been AMOs, or anti-microRNAs. And this is just a brief figure to show that the goal of these particular short nucleic acids is to target and bind and inactivate the microRNAs that are otherwise in this sort of reporter system would target the 3' UTR of, in this case, luciferase, and degrade that and lead to degradation of that mRNA. It just show this as a way to represent that indeed we're interested in other modalities besides, or for knockdown, targeting the microRNAs as well with AMOs. And what we've done with AMOs, and specifically in the case of end modification or 3' end modification with the 2' Mos, is tested this at various levels. So one of the great things about IDT, of course, is that we make the oligos, and so we're able to do these kinds of experiments and hopefully provide you with optimized design rules to go forward to, in this case, come up with the best modified AMO. And so what we see, kind of jumping to the chase here in the next figure, is that the full 2' Mo, it provides the best stability. We do see increased stability in, these are oligos that are incubated, I think, for 30 minutes in 80% human serum, which predominantly exhibits 3' exo activity. But these are the types of experiments that we would commonly do to evaluate different chemical modifications, in this case to be incorporated into antimers, but also the same would be true for antisense oligos as well. And we can see that ultimately the fully 2' methyl, if you look all the way to the right side of the screen, it's noted as 2' Mo, as well as with the 3 PSNs or 3 phosphorothioates at each end, provides the greatest stability. So these are, again, the types of experiments that we would look at when it comes to testing out these different chemical modifications. Finally, just to mention, and haven't really touched on it yet, would be the LNAs. The LNAs are, I think, known for providing the greatest stability. They are not as great at nuclease resistance, jumping down to the bottom there, but there is quite a boost, a 4 to 8 degrees Celsius boost for the LNAs. And they're not just used in antisense oligos, but also used for SNP discrimination in probes, most famously, I think. But you see once again that we've looked at different stability studies, and ultimately in application of some antimers, once again, the 3' end block with an LNA and a C3 spacer affords full stability of the particular delivered nucleic acid in this case, again in human serum. Additional deliverables, so I've just tried to highlight a few of our most studied nucleic acid offerings for RNA knockdown. I didn't show any functional data with AMOs, but showed some of the stability data there. You might also think about, including things like aptamers. We've done those types of development projects in the past. Ashley will talk about CRISPR reagents that could be, once again, encapsulated into nucleic acids, and those could be guide RNAs. They could be, I think, RNP complexes, although those have proven to be a little bit more of a challenge. And if you have ideas on how to best deliver with LNP, RNP complexes, that is the ribonuclear protein complex for CRISPR, we'd love to be able to talk about that more. But the Cas9 or the targeted nuclease can also be delivered in the form of mRNA or plasmid as well. And then finally, some of our synthetic biology reagents that could be delivered in this way and that we have delivered for different screening applications have certainly been plasmids and G-blocks. And Bob alluded to some things in conversation earlier that I think that the opportunity to deliver nucleic acid, period, whether it's a reporter system like a fluorescent probe, for example, can be thought about as well for these various types of applications. So with that, I'd be happy to take any questions you might have about the siRNA work that we've done or antisense oligo work that we've done.
If you identify a pretty active siRNA, somebody takes that into an animal model and shows that it works pretty well, What is the intellectual property situation if that person wanted to maybe take that forward to try to make a drug out of it?
That's a really timely question. In fact, so drug development for that particular, for our dicer substrates, for example, had been an exclusive licensure. I think I'm correct in this, exclusive licensure by Dicerna, who we did a lot of the screening work with from the time that I joined IDT in 2008 really till 2013 or 14. They have gone a direction in terms of their construct that has a tetraloop formation on the end. It no longer falls under the dicer substrate patent. And so I think the opportunity there for development is open for licensure with our dicer substrates. And I'm not sure about, you might also think about the Tuschl patented 21-mers. I'm not sure when those come off patent, but it probably is not too long from now.
Yeah, it would be good to get a pretty concise picture of that because obviously...
Yeah, no, that's a good question. That it is funny that it just came up a week ago. I'm not completely familiar with the IP landscape as we've gone a little bit of a different direction, but I do know that the development work that we had done under the Dicer IP patent is available now for clinical development.